Today, it's a great pleasure to have with us an extraordinarily learned scientist, Dr. Nutan Kaushik, Director General, Amity Food and Agricultural Foundation, Amity University, Noida, with us today. And it's really an honor. On behalf of the Director AFRI and on my personal behalf, I welcome you, ma'am, as our resource person for the monthly seminar for this month of April. Ma'am will be speaking on exploring phytochemical diversity of trees and medicinal plant species for their sustainable utilization. In fact, ma'am, I would also like to inform you that our director, sir, is not here today with us because he is traveling. There was a visit of the minister at dinner, so he had to urgently go there, and he isn't back. So, and we'll also be recording this session, and we'll be posting this today's webinar on our YouTube channel. The link will be available. We'll be sharing that link with you also. So it's an honor, first of all, to introduce Dr. Nutan Kaushik, though she actually needs no introduction. But her career is such that reading her this biographic sketch will be inspiring for the young scientists and the young researchers that are here with us today. It's really an honor to introduce her and to tell you about her scientific achievements. Dr. Nutan Kaushik did her Masters of Science in 1987 in chemistry from Agarwood University with specialization in organic chemistry. Did her PhD from IERI New Delhi in 1991 in agricultural chemistry. She has been associated with TERI for a very long time, from 1993 to 2017, and she was also part of as a faculty of the TERI University. She has been a DARD Research Ambassador. She has been a Cochrane Fellow at the IIA Michigan, USA, a DBT Crest Fellow at UC Davis, and the New Fic Fellow with the Royal Government of Netherlands in her earlier career days. Later, she has been a visiting scientist in major and well-known institutions around the world, ranging from Germany, Spain, Australia, Austria, USA, and many other countries. She has world-traveled and has a vast scientific experience. Dr. Kaushik has an expertise in metabolomics, development of new analytical methods involving volumetric, spectroscopic, and chromatographic techniques, etc. Dr. Kaushik is a professional with extensive experience of over 25 years in R&D and over 16 years in PG teaching in the field of biotechnology, plant protection, natural products, analytical chemistry, and agriculture policy. As head of R&D research group, she has been involved in project development and management, laboratory management, supervision and teaching of postgraduates, PhD students, and researchers, training of other researchers, and publication and grant writing. Dr. Kaushik has directed cross-functional and research teams using interactive and motivational leadership, acknowledged for her coaching, guiding, and mentoring team members to achieving resource-wise productivity and optimization. She has leveraged skills in analysis and analyzing collected information, comparing several types of research data, and engaged in safe research activities. They specialize in agro-industrial research, having worked on development of biopesticides and on methods of monitoring environmental pollution. She encourages researchers to develop skills, knowledge, and experience in aspects of research management such as budget and project management, teaching, publication and conference attendance, outreach activities, and membership of committees. Once again, a very hearty welcome to you, ma'am. We look forward to hear from you now, and over to you, presentation. Thank you so much.
Thank you, Dr. Tarun, for such an elaborative introduction, which is not required actually. I'll just share my screen, okay?
Sure.
Can you see my presentation now?
Ma'am, we can see you, but I think the presentation is loading.
Mm-hmm.
So we don't see the presentation yet. वो देखो साइड में presentation तो नहीं है उसको. वो presentation को वो करो नीचे. Pin करो presentation को.
Can you see now? I will stop sharing.
Ma'am, just a minute. We have some. उसको on किया. Presentation का है. अच्छा, ma'am, we could see the presentation. You can share again.
Okay. Now?
Yes, you can, ma'am. Definitely, we can see it.
Okay. It's okay?
Yeah, yeah.
Thank you. Good afternoon to all of you. Today, as Dr. Tarun was discussing with me what should be the topic for the presentation, so initially I gave the presentation on the bioprospection. Then he said that it will be good if we can change it to something related to chemistry. So this is the topic which I have given, like exploring the phytochemical diversity of trees and medicinal plant species for their sustainable utilization. So before I come to my main presentation, I'll just give a brief introduction about my foundation, because this is a new foundation created by Amity University. So this foundation, though agriculture has been a very long activity in at Amity University, but the foundation is a recent one. So we conduct research leading to excellence in the area of agriculture and allied field, including veterinary, animal dairy, and fishery sciences. We also work on dissemination of innovative technologies, imparting education in agriculture and its related field, contribute to quality human resources both to industry and educational institutions. Developing globally competitive human resources. So these are the broad goals of this one. And these are some of the key research areas in which the research is going on in Amity University under the aegis of this Amity Food and Agriculture Foundation. So like you can see, we work on like medicinal and aromatic plants. We also work on organic agriculture, nanotechnology, agriculture extension, so like you name it, and we are almost there for all sort of research activities. And we are open to new research areas also. Now coming to the topic, phytochemical diversity. As you all know that trees and medicinal plants provide lots of biomolecules which are useful as medicines, which are useful for agriculture, useful for cosmetic purposes, and also for the fuel purposes. Not only that, we also get textile, fiber, and so on. So biomolecules are extremely important when for the human beings right from the beginning of the evolution of the humankind. So like if you see the classification, we have been like there whole lot of compounds have been produced by like antioxidant, antibacterial, antifungal, anti-cancer, detoxifying agents, others, and they are basically belong to different groups like terpenoids, alkaloids, phenolics, polyphenolics, carotenoids, tocophenols, phenols, alkaloids, volatiles, and you name it, and the nature is there to produce it. The trees are there to produce it. The medicinal plants are there to produce it. And how they produce? They produce so beautifully without polluting the environment. They don't pollute any environment, rather they clean your environment and simultaneously give you these molecules. And so sophisticated molecules, such complex molecules which even human beings are unable to synthesize. You name this molecule called azadirachtin from the neem. So far the human beings have been unable to synthesize this molecule. So such a complex molecules the nature is producing, and such beautifully just by using the enzyme at room temperature, no high elevate in no factory, no smoke, no pollution, and rather they are using your pollution to create these biomolecules. So biosynthetic, there are three major biosynthetic pathways by which these biomolecules are produced by the plants, like shikmic acid pathway, pyruvic acid pathway, and the malonic acid pathway. These are the main three pathways, and you get the whole range of millions and millions of molecules produced through these. Whether it's a primary metabolites or whether it's a secondary metabolites, all the metabolites are produced using only this biosynthetic route. Now, coming to the medicinal plants, around 50,000 species has been used as medicinal purposes across the world. I'm not talking only about India, but I'm talking about the China. There are other countries like China, Brazil, and other where the traditional knowledge is very rich, and then around 50,000 species have been used traditionally. And China and India alone have the highest number of the medicinal plants used, like around 11,146 and 7,500 species respectively. So China uses about 11,146 and India uses about 7,500. And about 1,200 plant species are used by Ayurveda, Siddha and Unani system of the medicine. And 7,500 plant species are reported to be used by tribal communities. Our tribal communities has a lot of knowledge about these biomolecules. And apart from this, when we talk about your modern system of medicine, that is allopathic system, 25% of the prescribed drug, I'm telling 25% of the prescribed drugs are from the plant sources. Either they have been originated from the plant sources or they have been derived from the plant sources. So even during the COVID-19, the medicinal plants, these are very known spices which was used by a whole lot of people. Use this kara for preparation of the kara, the cinnamon, curcuma, holy basil, black pepper, munakka, and ginger. So during the COVID time also, the plants molecules served came as very handy in protecting us from the viruses. So, but what has happened because of the overuse? Population is growing, the demand is growing, and we have been doing the unmindful exploitation from the natural resources. What has been done from the forest area, from the wild areas, we have been collecting them without, and which has created threat to existing flora and fauna. So there's a need for the cultivation, restoration of the natural. So on one side, we have to cultivate them, we have to develop the practices for the cultivation and domestication. On the other side, we also have to restore the natural habitats. So, but I see a lot of opportunity here. We see there are a lot of opportunities, particularly we are so blessed that out of 25 biodiversity hotspots, 4 are in India, and so we are blessed with these 4 biodiversity hotspots. It means there are species which are endemic to these areas and they do not occur anywhere else. And moreover, after this pandemic, not even before the pandemic also, there was an interest regrowing because of the allergies and so on from the allopathic system. But particularly after the pandemic, people are coming back to the natural remedies. So renewed interest in traditional system has increased the demand. And then, but if we really look at the number of plant species which are there I think in our nature, but only 3,000 plant species have been investigated scientifically. So there's need to do the scientific validation, even of the traditional knowledge, and then come forward with the new molecules, new formulations, and so on. But these metabolites production is affected by several factors. One is the season. Season affects the concentration of these metabolites in the plant material which you are collecting. The physiological stage of the plant or the tree affects the concentration. The geographical location affects the concentration. Environment in which it is growing affects the condition like temperature, humidity, and soil. And then harvesting time also. Harvesting time is directly linked with the physiological stage, and then the genetic variability. So all these factors contribute to the variability in the concentration of the metabolites which is present in a tree, in a plant. Like plant contains a metabolite, for example, A, B, C, D, and E. But the amount of this metabolite which is present in the plant can vary from season to season, from physiological stage, from location to location, to temperature, with the effect of temperature, harvesting time, and then because of the genetics. So, so this is the approach we basically. So bioprospecting is coming up in a big way for exploring your plant diversity, the plant diversity which is available for as a source for secondary metabolites or the new genes. So, and this is the approach in biochemical prospecting which we follow. We classify the plants into two types. One is the plants which has an active known molecule, where the known molecule activity of the molecule is known, or the molecule is known, like this Artemisia annua, Vidania, and so on. So we prospect the plant with respect to the diversity of the active principle, the active biomolecule which is present in this plant. So we screen these plants, different populations, for the amount of the metabolite which is present in with respect to, and optimize and the variety of the plant which can give you the highest yield of the secondary metabolites or give the highest efficacy at this time in different plant varieties and accessions. Second type of this bioprospection is with unknown active molecules. There are many traditional knowledge like where we do not know what is the bioactive molecule which is responsible for a particular activity in a plant. So here we go for the prospection of the new active, identify bioassay mediated isolation of the new active molecules we go for. So this is the basic approach which we follow for known active molecules. We collect the germplasm or the population or the plant samples from maybe it could be leaf, it could be bark, it could be stem or root, depending upon where the activity is reported from. And then we quantify the active principle with the help of the GLC and HPLC, and identify the elite plant in terms of the active principle. Then that elite can be multiplied and then through tissue culture, and then we go for the cultivation. And after the cultivation, then with the help of this will help to conserve the biodiversity by sustainable harvesting to meet the demand of the raw material. So I'm unable to change my slide. So yeah, I could. So the active the active amount of the active principles is important in the raw material because that also determine the market rates. For example, solenoside, solenosodine in Solanum cassia determines the price of the raw material. Similarly, taxol content in Taxus baccata, methanolites in Methania form, somnifera, artemisinin, tigrasin, azulene, pyethins, and so on. There are many examples. This is the approach which we follow for the prospection of the plants with unknown active molecules, where the activity is known but the molecules are not known. So we collect the plant part and then we prepare the extract, do the bioassay, and from the bioassay we do isolate and identify the active ingredient. And after the identification of active ingredient, we go for the filing of the patent, and then we proceed with the preparation of the appropriate formulation, go for the formulation trial, toxicological evaluation. It's very important to do the toxicological evaluation because you never know if the whatever the molecule you are developing should not be harmful to the human beings or to the environment. So that way we have to do the toxicological evaluation, then the screening of the elite plant, and the multiplication and DNA fingerprinting of the elite plant, and then we go for the commercial exploitation. So these are the total approach which we follow. So coming to the bioassay, we can identify what type of activity we are looking for, whether we are looking for antiviral, we are looking for antimalarial, we are looking for anti-cancer, fungicidal, insecticidal, or depending upon that bioassay is done and then we standardize the bioassay and we use GCMS, HPLCMS, preparative, HPLC, NMR, etc. for characterization. Now I will give you some examples, the application part of it, like some of the projects which I have done in during my career. So this was a project for the prospection of the chemical diversity of neem growing in India for value addition. This was a Agarwood project which I did. I was the nodal of national coordinator for doing the biochemical evaluation of the entire germplasm which were collected from the entire India. So this was, so if we really see what are the biochemical constituents of the neem, so we have the azadirachtin, which comes in the seed cake after the extraction of the coil, and which is used by the pesticide industry. Is the oil, which is also used by the pesticide industry as well as by the soap industry. Oleic acid, which comes in the oil, is used for oil stability. Nutritionally desired linoleic acid is an essential fatty acid. Stearic acid is used for margarine and shortening. So, as I told you earlier, that biochemical composition is affected by genetic factor, environmental factor, time of harvest, and quality of seeds. So, individuality varies in composition. So, before this network, some studies were conducted, but there was no extensive study. I'll just skip this one. So, there was a partnership was built up between 11 institutions in this program. So, there was AFRI, KAZRI, AFRI, HU, IFGTB, IIT, NVPGR, NVRI, TERI. So I was in TERI at that time. So I was a national coordinator for the collections done by all other institutions. While NVPGR, they did this deposit. They conserved the all the germplasm in their gene bank. So this is the coverage of the collection. You can see the almost entire India was covered under this network. And then this is the number of collections which were done in this network project, so from different states. And this is the variability, lot to lot variability, which we got from in terms of the all these parameters. So I'll just go for this overall variation. So if you really see the azadirachtin content, it's varied from 0.02 to 1.6%. So what does it mean? What does it have? Does it really mean anything? You can have 1.02 what's of fun, but it really has got a meaning. Like if you're looking for the bioactivity, if you're using the seed for biopesticide industry, and if you are preparing the extract from 0.02% as a containing seed, then you're not going to get any efficacy. But if you're at the same time, if you're like sourcing the seed which has 1.5% azadirachtin content, definitely biophase is going to come. Same is true with the medicinal plants also. If you're preparing your kada from the one which does not contain right amount of active ingredient, it is not going to work for you. Whereas if you're going to use the one which is having the high amount of the biomolecule or the active principle, it is certainly going to have the impact on you or effect on you. It is certainly going to control the disease. That's why before this network, what was happening, that some people used to say the neem extract, if we prepare on field, it works. Some used to say if we prepare neem extract on field, it doesn't work. And the reason is this variability. So it depends what type of seed you are using. Similarly, for the oil content, 24 to 56%. If you are looking for the trees for the oil, then you should go for the trees which are having the 56%. Try to go for the 24%. Because in industrial application, these variations are huge variations. Even for the industry, even 2% variation is a huge variation. It like impacts their economics quite significantly. So similarly, oleic acid 32 to 68%, palmitic 6 to 46%, linoleic acid 3 to 36%, stearic acid 0.5 to 26%. So these are the variations which we observe. So as coming to the azadirachtin content variability, so this is the state-wise variation. So we divided this population from different states, and you can see the seeds coming from the southern plateau have the high azadirachtin content, where the area which has high temperature the azatic content is pretty low. So this is the like you see, for Andhra Pradesh has the highest, then the Karnataka, then Tamil Nadu, Delhi, Chhattisgarh, and so on. So azatic content was found to be greatly affected by seed quality. Also by seed quality, seeds having very poor quality, that is high fungal infection, showed very low azatic content. We did use of fungal infected seeds in our study, but this was an additional study which we did. So sometimes what happens from the top, if you see the seed, it looks pretty good. However, if you break this seed, you can get this fungal infected seeds. So this is the one which is not good for the quality. So you should not go by just by the look of the whole seed. You should also look for this. You should break the seed and see. This happened particularly when the moisture content is high during the storage. If the moisture content is more than 10%, then you will get this infection, or you have stored the wet seed directly without doing the proper drying. Then you get this fungal infection very easily. So I used to get lot of such type of seeds which I used to reject. And then sample having this, these are the few accessions like plant which was identified as having more than 10,000 ppm, means more than 1% of the aza content. So you can see very few out of 1500 accessions, only this 10 accessions contains more than 1% azatic content. And then samples containing 1000 to 7500, also very few, not many. And then sample containing, So, and then oil content variability. So this is, you can see, now it is almost reverse kind of average oil content. So this is the Pareto chart of this oil content around, it was a uniform. So large number of samples recorded oil content between 45% to 50%, and seeds having high moisture content recorded poor oil content, less than 30%. So these are some of the samples which recorded more than 50% oil, and then these are samples having. I think it continues up to second slide. Then fatty acid variability, you can see there. You can see from state to state there was a variability in different fatty acids, which was like estimated in these accessions. So sample from Maharashtra, Delhi, Haryana, Chhattisgarh, Rajasthan, Gujarat have high oleic acid, more than 50%, and seeds collected from Tamil Nadu and AP have comparatively higher palmitic acid and lower oleic acid. So this is the distribution of oleic acid variability, and then this is stearic acid, palmitic acid, and so on. So this variability can be used to study the diversity and also can be exploited for industrial purposes. And after this network program, what happened? The industry started giving started paying high price to the seeds coming from the southern region for the azathiotic content, because it was recording high azathiotic content and high oil prices to the oil coming from the northern region. So this was the impact of this project, not only in identification of the elite planting materials, but also it has very policy implications in terms of the remuneration which the collectors were getting. So performance over the year, we also studied the effect of from the same germplasm, from the same tree, from the same plus tree, the seeds were collected into continuously for 2 years, and we didn't find any much variability in terms of the oil content. In terms of the azathiotic content, what we found, the effect of there was an impact on the azathiotic content, but the relative increase or decrease was same with respect to the samples. Suppose sample A contained 1% azathiotic content in first year, in second year it contained 500 ppm, and there was a reduction of 10%. So in all the samples, the reduction of 10% was recorded. It's not that only one particular sample got reduced. So it was with respect to oil the population. So this is again, you can see the azotating content. In first year it was low, in second year it was pretty high, but high increase or decrease was with respect to this thing. Similarly, palmitic acid, and then out of this project we got this publication in Current Science, and then this is with Dr. Tomar for the Gujarat samples, again paper with Dr. Tomar. And similar type of project was done was immediately after the Neem network. There was a Jetrofa network because there was a whole lot of like what you call hype was given to the Jetrofa. That Jetrofa is the tree which will which will solve the problem of the biofuel. So as a it was being promoted as a biodiesel tree. But nothing was known about Jatropha by that time. Nobody knew what is the yield, how much accession, how much variability we had. So this Jatropha networks were created by initially by Novood board, later also by DBT. And fortunately, I was the national coordinator for these two networks, Novood board and DBT, and I analyzed around 3,000 accession collections for this thing from this, but there was not much variability was observed. So, but we could identify the population into high oil content, more than 35% seed oil, or moderate oil content, 30 to 35% seed oil, low oil content, less than 30% seed oil. And then we did this multi-location trials with the high oil yielding plants, and then we could identify the plus. But however, Jatropha could not perform well in terms of the seed yield. So it's still being pursued, but not so seriously. So it around 2,500 germplasm were analyzed in DBT network, and around 500 germplasm were analyzed from Novood network. And more than 300 trees with high oil content were identified. This was another project which I did with from DBT, which was on chemotyping of the Vidania somnifera. I think everybody knows Vidania somnifera. It is ashwagandha, and then it contains a lot of vithanolides. So this project was to study the variability of this vithanolide in different collections from different parts of the India, and then assess its variability and identify different chemotypes. Because Vidania contains three chemotypes. There were three chemotypes which are already reported in Vidania by the Israeli group. So, but nothing was available for the Indian accession. So I collected around 162 accessions of Vidania subgroup from different states of India. We analyzed. So after we collected the seeds, because it contains a lot of air, because the time of collection was varying for different accessions. So we collected the seeds, we grew them in our field simultaneously, and then we did the analysis. And what we found, and then we developed the method of estimation, and we identified the chemotypes and serotypes. So this you can see the field here. You can see this is the wild type. The tall one are the wild collections, and the dwarf ones are the one which are cultivated varieties. So we collect germinated all the germplasm in our field, and then we collected the leaves in different at different physiological stages and analyzed them with afferin A content and with anolite AD variability in these successions. So these are the three chemotypes which are already reported from the literature. So according to this literature, this chemotype 1 contains vidoflurin A as a major constituent. The chemotype 2 contains vithanolide D as a major constituent, and then the vithanolide E contains as a major constituent. So when we analyzed our whole germplasm, we could divide the germplasm into chemotype 1 and intermediate of the chemotype 1 and chemotype 2. So all the collection from the Mandsaur, Yamunanagar and Noni, they belong to the chemotype 1 having only the vidoflurin A as a major constituents. Whereas the other population from Bhopal, Gopeshwar, Jaipur, Delhi, Jodhpur, Udaipur, Chandigarh, and so on, they belong to the intermediate of chemotype 1 and chemotype 2. If you easily see the chemotype 1 and chemotype 2, chemotype 1 contains vitaferin A as a major constituent, and rest of the peaks are small. Whereas in chemotype 2, the light D is a major constituent, and rest of the peaks are small. Whereas the one which we got in our case, this is the chemotype 1, clear-cut case of the chemotype 1 having only vitaferin A peak as a major constituent, whereas the other one contains both the peaks, vitaferin A with the denominator D with equal height. So it means they were the intermediate of the this chemotype. Then we investigated the effect of plant growth stage on the withanolite content. You can see here, I'm just giving the data of the 3 hexagons, though we have the data for the entire 162 hexagons. So these are the different physiological stages. One is the vegetative stage, reproductive stage, and the harvest stage. So you can see during the reproductive stage and the reproductive stage, the withanolite content is more or less the same, although the vegetative contains the highest, but it's more or less. But it drastically reduces at the time of the harvest. So we thought maybe from the leaf it is going down to the root, because we normally use the root. We don't use the leaf in the Ayurvedic system. It is a root which is traded. So next time what we did, we estimated the withanolite content in leaf as well as in the root simultaneously. And then here you can see, no, it was not the case. It was not being translocated to the root. Rather, we see the decrease in the root and as well as in the leaf simultaneously. If the withanolite content was decreasing in the leaf, it was similarly decreasing in the root, simultaneously decreasing in the root as well. So there was no translocation of the withanolites from root leaves to root. So I was wondering, like, whether is it my question always which I posed is that is it really withanolites which are responsible for the activity? Because traditionally we have been using the root, and root doesn't contain that much of withanolite as compared to the leaf. So what is it which is contributing to the immunomodulatory properties of the vitaferin? This is still a question mark. We still need to investigate this. So this is the range of vitaferin A content which we got in different accessions at different physiological stages. So you can see it varied from the mean content was 1.09, 1 during the vegetative stage, 1.2 during the reproductive stage, and only 0.5% during the harvest stage. Whereas in the root it was 0.12 during the vegetative stage, 0.59 during the reproductive stage. It was and it was 0.02 during the harvest time. So very minimal amount. We also did this variability of the polysaccharide content. So polysaccharide content was varied from as low as 0.2% to as high as 50%. So maybe these are the polysaccharides which are responsible for the immunomodulatory properties. We still don't know. Nobody has worked on it, so we don't know about it. So variability with the berberin content in Berberis. So another plant which I worked on was the Berberis. So we studied the variability of the berberin in various collections, and then we could see this leaves. We could see the 1 point in Berberis. Berberin is present in the root. Basically, it is the root which is used for the Berberis extraction, and then we could find out the variability in the root as well as in the leaf. So this is the variability in berberin content. You can see in different species: Biosynthetic, Lycium, Gythria, Tinctoria, Umbretta, and Aristata. So from different population. So this was also quite significant variability. So maximum average content was obtained in root of B Lycium or Dehradun, 2.9%, followed by the root in Biosynthetic and Champawat region. So another plant which we studied was the Seabuckthorn. Seabuckthorn, the fruit during the different ripening stages, what is the biochemical changes which are occurring. So we collected the fruit in 4 physiological stages: phase 1, phase 2, phase 3, and phase 4. And we studied the variability of the different component. And what we found, that if you're targeting the lauric acid as your fatty acid content, then you must harvest it at early stage. But if you are targeting the vitamin A, then you must harvest at the late stage. So stage of collection or picking of the fruit is very critical when comes to the biochemical composition of the fruit. So this is another project which I did, the Indo-German project on the Tylophora indica. Dr. Tarun, how much time do I have?
Ma'am, you have enough time. You can keep continuing. We have time.
Okay. Yes, fine. So screening of phytochem, this time is until 5 or 5? 5:30. My time is until 5 or 5:30.
We have kept last few 15 minutes for some question answers. So, ma'am, you can safely go up to
5.
Say 5:15. You can continue.
Okay, fine, good.
Yeah.
Sorry. So now coming to this, this is another project which we did. It was an Indo-German project. Like Tylophora indica, everybody knows this is the root is used for this lung disease, and then it contains tylophorid alkaloids. So this was so we did this. We isolated these compounds, 4 compounds, pure compounds, 1, 2, 3, 4, 5, and then we developed the Hplc fingerprinting of this Tylophora indica to study the variability of these alkaloids in the collections which we got from northern India as well as for the southern India. And this is the typical UV spectrum of these alkaloids. It contains, it has a lambda max at 258 and 286. So this is which can tell you that this your sample contains the tylophorid alkaloids or not. So this is again the same chromatogram. So we collected the germplasm from different places. You can see this is the variability in the leaf shape, and then alkaloid concentration also varied quite significantly in different samples. So we also developed the Hplc Msms profiles of these ones. So you can see to our display what happened. Like the excisions which were collected from the south didn't match at the Hplc LCMS profile of the from the southern region. Excisions didn't match with the excisions which were coming from the northern region. From the northern region, we were getting this. You can see this molecular weight of 409. 409. Here you can see the biggest peak contains the molecular weight of 409, and 409 is not the molecular weight of the tylophorin. It is the molecular weight of the N-oxide. So it is an N-oxide form of the tylophorin which were present in these samples, not the pure tylophorin. This was revealing for us the very interesting fact for us. Like all these are the forest area from near the south in the Mysore and the RD Halli and the RD. This is the second sample. All the samples they contain 409 as a molecular weight. So we were really very curious to see that we did this analysis of sample from northern region which came from the cultivated area, and that perfectly contained the molecular weight of our tylophorin. It didn't contain the molecular weight of 409. So this was a revealing part from as far as the tylophorin content diversity was concerned. Then we also studied the bioactivity of these Tylophora indica. So we contain 2 points approach: the identification of active extract and fractions by classical methodology, and also by the Martin method. So this is a classical approach where we had this ethanol, and then we partitioned and we got certain fractions by partitioning, and these are the fractions obtained by partitioning. And then you can see the fraction 2 contained very good activity at almost 155 times higher than the original ethanol extract. So you can increase the activity of your extract just by doing simple partitioning. This is what I'm trying to emphasize here. So you can remove the unwanted material from your extract, and then you can enhance the efficacy. This is the feeding inhibition PIOSA which we did against the Helicoverpa larvae. We can here you can see this EI50 value is only 5.22 ppm and 2.82 ppm, which is pretty high. And then this is the against the moth oviposition dead trend. It was quite dead trend for the oviposition of the moth of the Helicoverpa arbigera. And this these are the like 4 alkaloids which we isolated: tylophorin, isocerebrin, tylophorin, tylophorinidin. And then we did this efficacy of the pure alkaloids also. You can see this azathioprine was the positive control. So azathioprine recorded 0.7 ppm with this EC50 value, whereas the tylophorin recorded 1.5 ppm EC50 value. So it was pretty effective. All the alkaloids were effective. So then we developed the formulation of this tylophora, and we did the field evaluation. So this you can see the larvae. This is the control. The big one is the control, and the smaller one is the larvae which we obtained from this treated with formulation. So this is a cotton crop which we studied. Here you can see we also got the significant increase in the yield of the crop. Not only the control, but also control. So after this Tylophora indica, I did an Indo-French project with Professor Dubois. Professor Dubois is an expert in saponin, and chlorophytum contains a lot of saponins. So before we started this project, the chemistry of this chlorophytum species Borivilianum was not known actually. People did not know what are the type of saponins which are present in chlorophytum Borivilianum, which is mainly used in India. So we submitted this project, another French project, for isolation characterization of the biologically active saponins. So what we did, these are the different species which are available in India. I think you people know better than me. So this is already cultivated Chlorophytum bolianum. So this is the crop, then you have the plant, you have the root clusters. Again, it is a root which is important in the Chlorophytum borivilianum. So we did this root saponins was prepared, and then from that we did the vacuum liquid chromatography. We obtained 3 fractions, and from the methanol fractions we got around 7 new borivilianoside. So we named them borivilianoside A, B, C, D, E, and F up to H. We named them, and this is a published paper. You can find in Journal of Natural Products and so on. So this was for the first time that these borivilianosides were isolated and characterized from the chlorophytum species. So we isolated almost 14 new compounds, 19 pure steroid saponins from Chlorophytum borivilianum and C. Chlorophytum orchidestrum. And as we also isolated 5 pure compound from MCB 405. MCB 405 is a variety which is being cultivated at that time in Mandsaur, and not only Mandsaur but from Udaipur also. I think we got it from Udaipur, MCB 405, and we isolated 2 compounds from Chlorophytum glycosides and 3 compounds from Chlorophytum coloporeanens. So this is the structure of the different Chlorophytum borivilianosides which we isolated. This has the aglycone, which is an steroid, and then these are the sugar attached with this one. So borivilianoside D and H, 5A, 6A. This is from the orchidastrum, the compound from orchidastrum. And then we did this cytotoxic assay of this borivillanocyte. And then we could find the activity, but not as high as the paclitaxel, which was the positive control. So but the paclitaxel contains in nanomoles activity, how the activity which we recorded was in the macromole. So borivillanocyte recorded some kind of cytotoxicity, but not that great as the paclitaxel. Then we did this insect bioassay also of these borivillanocyte. We got some good activity with borivillanocyte C with IC50 value of 0.7. Okay, it is the IC50 better than the Neem. So it is the IC50 value which is important when we talk about the bioactivity. Nothing else but the IC50 value. So you should concentrate when you say the activity. You must concentrate on the IC50 value. What type of IC50 value you are getting? It's not that it is effective and at what dose. It means it will determine the dose and commercial viability of your compound or the molecule. Then we also developed the Hplc chromatogram, which were not developed prior to our studies for different species of the Chlorophytum borivillianum. This is previously was the borivillianum. This is the again the Caesia glaucois, Chlorophytum glaucois, then Nagpurensis, Colapurensis, and then different species. We developed the Hplc chromatogram. Another plant species on which I worked was the eucalyptus. So eucalyptus leaves are used generally for the oil extraction. After the oil is extracted, the leaves are not used for any purpose. They are generally used for the combustion for the what you call for the in the for energy purposes, for utilization as energy process. So what I did, I prepared the extract from this oil from which the essential oil was already extracted and prepared the different fractions, and we got very good activity in this one. And we isolated different compounds from this one. So ellagic acid was found to be responsible for the growth inhibition of the larva, and then bethylenic acid was found to be responsible for the mortality of this one, and then the fluoroglucinol for the antioxidant activity. So it contained three types of activity: growth inhibition, mortality, and antioxidancy. These are HPLC chromatograms of the fluoroglucinols which we got from this eucalyptus leaf extract. And this is the mechanism by which it was controlling. You can see in the control, the cut is clear, whereas in this eucalyptus treated insects, the gut has thick. There is a thickening of the gut, because of which the insect was not able to absorb the nutrient, and it died because of the lack of nutrient, because it was not able to absorb it. And then we developed the formulations, and then we did the field trial, cotton and chickpea, and then we also did the toxicology just to see whether it's really toxic to human beings or not. It was found to be absolutely green, and so this was the formulation which was developed and has been registered in India and has been given to 3 companies with Polcure, which is effective against cotton bollworm initially, but later on, when it was given to the American company, they found it effective against 30 more insects. So this biopesticide has been successfully developed and commercialized. It has. Then this is another project which I did on agarwood. I think you people all must be knowing, being a forester. Agarwood is valued for its agar content, with the black agar which is being extracted from the dead wood, hardwood or the trees, and this occurs because of the infection inside the tree. And naturally, the infection occurs. Infection may or may not happen inside the tree, and if it happens, it may take 30 to 50 years to get the full infection inside the tree. So this was the DBT supported project, where what we did, it is known that this agarwood infection occurs, agarwood formation occurs as a response to the infection, either due to some uh, microbial agent or due to physical injury. So what we did, we isolated the microbes from this agarwood tree, and then we re-inoculated those microbes artificially inside the tree species. And then you can see here, these are the microbes. And then after reintroducing this microbe, after reintroducing this microbe, what happened? Within the 3 months, we infected this 5-year-old tree, and within the 3 months, we could see the development of this infection. You can see the dark brown patches over here. We extracted this wood and did the GCMS, and we found that it was very well producing the agarosperol, which is responsible for the fragrance of this agar. So this paper is published in Frontiers in Microbiology. So we isolated around 1,000 microbes from this tree, and then we tested about 15 microbes, and we found 3 microbes to be very active which was very active in inducing this infection production of the agarwood, and this technology has been tested in the ground in the field, and then we are doing a pilot testing of this one. This can be taken up on larger scale for livelihood enhancement. Particularly, I think now the in the agarwood is also being now be cultivated in southern part of India. It is being introduced. So this is another technology, biomolecule, but of different nature. It is a highly valued molecule. I think it's costlier than even gold. One tola of oil is equivalent to, I think, 1 lakh rupees or something like that. I don't know, but it is highly expensive and very high demand in Middle East. So thank you very much for this one. But this here I would like to end. I had more slides on the quality control, HPLC fingerprinting, but I thought this let me focus only on phytochemical diversity this time. So this is Professor Peter Proksch on the first photograph, with whom I did the Indo-German project on the Tylophora indica. On the right-hand side is Professor Du Bois, with whom I did the Indo-French project, then the Indo-Spanish project, Dr. Azucena, and in the left-hand side you can see the Tylophora vine. So with this, thank you so much. We cannot hear you.
Thank you so much, ma'am. Yeah, ma'am, can you hear me?
Now we can hear you.
Yeah. Ma'am, it was in fact really a wonderful presentation, a wonderful talk by you. And here, though you can only see me, but here there are around 25 people sitting with me, all my colleagues here. Different scientists and officers are sitting here. And everybody has been, enjoying and listening very carefully to, the magnificent work that you have done. And it's really, I should say, kudos to you that you have worked on so many species, ranging from Neem, Jatropha, Withania, then Tylophora, even Agarwood, and also Safed Musli. And it's really very heartening to see that for the first time the Borreliosides that you had for the first time identified and characterized, which is a very big thing. To identify unknown, because as you were showing in the beginning that there are two ways: either to see the ones which are already available, the metabolites which are known, but to work with the unknown ones. I think that is the most challenging, and I think that is also the beauty of this work, that you find something totally that is not known to the human beings. So finding that out, characterizing that is something big, and it was really so nice to have listened to your such a rich lecture, and the mega work that you had done on so many different aspects. And I know, there are so many other things that you haven't yet told us. But before I tell the others to ask questions and we open the question answer session, It will be really, in fact, I'd like to tell the House here and to all the participants, the association that we have had with Dr. Kaushik is not new. As she has already indicated in her beginning slides, she has been associated with the Novode board project. She was where we had a big contingent from ICFRE also working in the project. And that was a big project which, in fact, laid the foundations to a lot of Neem research that is going on in the country right now. And interestingly, right now, I'm also the national project coordinator of another Neem project that is going on right now. And very interestingly, Kaushik ma'am is again part of that. Recently, the director AFRI and Dr. Kaushik has signed an MOU. So Amity and AFRI have entered an MOU recently, and that MOU will be is as a part of the AICRP 26 on the genetic improvement of Neem. And under that, I'm glad to inform you all that under that MOU, under the auspices of that MOU, uh, uh, ma'am will be actually carrying out the azadirachtin quantitative estimation from the neem seeds that will be collected from the CPT selected from 10 different agroclimatic zones that we are doing under a national project. It's a national coordinated project having 6 institutes. So around the country, we are again doing a fresh analysis. And as ma'am has already pointed out, that in Neem, it was found out that the ASA content is not very stable. Sometimes the same accession may have high ASA, sometimes it may have low ASA. And not just that, the South Indian accessions had higher ASA compared to the North Indian accessions. Maybe high temperature was detrimental. But what I was wondering was that it is the azadirachtin is a secondary metabolite, and usually, as it happens with secondary metabolites, a lot of compounds that under the extremes of weather condition, there is more synthesis of the compounds under different biotic and abiotic stresses, whether it is heat or it is drought condition or maybe some insect attack. These metabolites are secreted at a very large extent. So it was really surprising to see and to also I've been reading all these papers under as being part of this Neem program now. What I'm surprised to know is that why Rajasthan in the hotter climatic condition accessions were having very low ASA, which is very unlikely. I could not really find the answers to it. So what I was thinking was maybe there is something else that might have happened. It's just a theory right now because I was wondering because trying to work out the logistics of this project that we are trying to steer right now. I was thinking that maybe during the course of shipment of the seeds collected from Rajasthan, by the time it reaches to the analysis part, maybe there is already a decay because the shelf life of azadirachtin is very, very low. Because if not properly handled at high temperatures, ASA actually what I have also come across is that under cold temperatures in slightly acidic medium, ASA is better stable. There is a half life of 50 days of azadirachtin. But here, maybe due to high temperature, and that is also under temperatures of around 10 to 15 degrees. Now we are dealing with 40 degrees, 50-degree temperature here in Jodhpur and in the regions in Rajasthan. So maybe we are losing azadirachtin. Maybe the contents are high, but we are losing out on azadirachtin because of poor handling. Is that the case? Can that be the case, ma'am?
Oh, I don't think so. Only because of the handling,
I'm sorry,
it
we
could
cannot
be.
hear you. Yes.
Sorry. I
Yeah,
don't.
we can hear you now.
Yeah, I don't think so. It could be on handling could be the only answer. So this I don't because it was
Okay.
done not only few samples. It was done across the samples, many samples. And if we talk about
Okay.
the quality of seed, we only took the best quality of the seeds only for analysis. If the quality was inferior, we didn't take it forward for the analysis. So this was the criteria. Only the best of the best
Okay.
quality seeds were taken
Right.
forward, and then it was dispatched immediately after the harvest. The person it was personally, not by even by post. The seeds were dispatched personally,
Okay.
and they were kept in a 24-hour air-conditioned lab.
Okay.
So the storage was at proper storage. Storage was in air-conditioned lab for 24 hours. We had the 24 hours cooling in our lab. So the storage was done in those conditions,
Okay.
and then it was immediate after. I think within a 2 days of the harvest, the seeds were transported to us personally.
Okay. Okay.
So,
Okay.
but
So ma'am,
but you
actually,
have a point.
since we are,
But you have a point. This can be investigated. The effect of temperature
yeah,
on azorectin variations, the storage conditions,
yeah.
the effect of
Hmm.
temperature. It has been studied, but I think there's a paper, if I remember correctly, there's a paper on it on the storage conditions.
Yeah, actually, I have read that paper, ma'am, and that paper says that high temperature is not really good for azorectin storage,
Hmm.
and that is what I mean is also because there is in the market also so many formulations having azorectin is available, but when we try to use them, the efficacy is not that high because maybe the standard operational procedures SOP is not properly maintained by many of the
But
industries which are I mean
that might be one thing. Second thing, the formulation when it is approved, the CIBRC asks for its shelf life studies of the formulation at different
mhm.
temperatures, at different three
Okay,
different temperatures, and one of them is the room
okay,
temperature also. Unless that is
okay.
it is effective, it is efficacious, found to be less than 6 months, then it is not approved by CIBRC.
Okay. All right. All right. In fact, ma'am, this is because I was just concerned because now this is a project we are running. So from, I mean, there must be some learnings from your past
Previous.
projects. So I mean, you can probably also share with us what we should do and what we should not do while collecting the seeds, so that I mean we can have
I
a
would
message
suggest
across
for
the
collection.
partners.
I would suggest for collection you
Yeah.
must conduct a training for all the partners, and Dr. Gurdev Singh is the right person for
Okay.
conducting the training. He has
Okay.
developed the
Okay.
method for collection of the good quality team seeds. He's the one.
Okay.
So he has developed the protocol.
Okay. Okay, so I'll try to arrange sort of a webinar with him also, and
Yes.
I'll try to connect all the partner institutes so that we can have a sort of something from him. He can tell us,
Yes. So he is the right
Dr.
person
Gurudev
to
Singh.
conduct this training, how to collect the good quality seeds. So this is the right time
Okay,
that he should conduct this one. He must be having some videos and
okay,
something through which he can show it. And if I have
okay.
some video of his training, I will share with you. And
That will
from
be great. That will
yeah,
be great. If you have something you can share, and
yeah.
that will be a really good resource for us.
Hmm.
Yeah, and ma'am, moreover, one more thing, particularly about the Neem thing, before I, in fact, ask others also to ask the questions, is that as you said that you were also trying to show us there was quite a lot of variation in terms of like the South Indian Neem and also the Neem exceptions from the North India. But when it came to oil, I found that the Rajasthan accessions were having very high oil content.
Okay, welcome.
So, ma'am, my question is that how much, first of all, was there a positive correlation between ASA and oil content? Is there at all a correlation between the same accession having, I mean, same,
This is reverse negative, reverse correlation.
there was no correlation.
Inverse
I mean, so
correlation. So if the ASA is
inverse
high,
correlation.
oil is low. Oil is high,
Okay,
ASA
okay, so
is
there is an
low.
inverse correlation.
Yeah,
Okay, inverse correlation.
inverse.
So if we have to do selections for a genetic improvement program in which if we do the selections for ESA, then we will have a different set of plants,
Different
and
set
for
of
oil we'll have different set of
different set
plants.
of plant. Altogether different set of
Okay, okay.
plant.
And ma'am, and how much do you think is the genetic association, genetic part in terms for ESA, and how much is the environmental contribution for the ESA content?
Environmental contribution is certainly very high because, as I showed you, from year to year there was a huge variation in the as a content. However, the
Yeah,
superior plant remains superior even the year one in the year two. So
right.
relative superiority will always there, but the content per se is directly affected by the particularly with the rainfall.
With the rainfall.
With the rainfall is the highest
Okay. So,
correlation.
means ma'am, high rainfall has a positive correlation. High rainfall has a positive correlation.
Yes, high rainfall has a positive correlation, and
Okay,
then low
okay.
rainfall has a maybe that is the reason that Rajasthan has a low as a content.
Yeah, that also makes sense. Definitely. Yeah, yeah, yeah. Yeah, thanks, ma'am. Thank you. In fact, I'll be in touch with you along the course of running of this project that we are right now doing. So I'll be having many more chances to interact with you and we other team members also. Probably we'll also be organizing. seminar with you with the AICRP 26 Neem team. We call it Team Neem. So the Team Neem members, all the 6 institutes will be joining with you maybe during the course of this year itself, sometimes 2 or 3 months down the line. We'll have a meeting with you once again.
Sure, sure.
But I won't take much time. I'll just, the house is now open to others to ask questions. If any of the participants here who are sitting offline with me here in the seminar hall, as well as the online participants. So we first open this for the offline participants. If the people sitting here in this seminar hall, if they anybody of you have any question, you're most welcome. Particularly the people who are already working on botanicals, people who are working on preparation of insecticides, botanicals. You are most welcome. People who are working on entomological aspects, you people can have questions. Protection division people, if there are any questions. And I would suggest that you can come here on my side here, and because the camera has a not a very high range, so you can come to the speaker and ask the question here. Anyone?
Madam, is there any relationship between tree age and oil content of this tree?
No,
Azadirachta
no,
indica.
no,
There is no relationship.
no relationship with age. Yeah, the
Okay,
oil content initially for 5 years, yes, there's a slight, uh, uh, but after 5 years, it stabilizes.
it is stable.
It
All
stabilizes
fine.
after fifth year.
Okay.
It stabilizes.
Okay. Thank you. Anybody else? Anyone? Uh, Shivani, do you have a question? You can come here. You have to come here. Sapna, do you have a question? You can come here as well. आप लोग इधर आ जाएं बस कैमरा.
Good evening, ma'am. Ma'am, actually, I want to ask if we are like working on botanicals and we are able to find out what particular AI in that particular botanical is. So after validating it that yeah, it has potential, then we have to go for formulation and then we have to register it. So ma'am,
Yes.
before registering it, we have to test it on I guess mammals, rabbits, and rats. So, ma'am, for that, what is the procedure like? What we have to do?
Okay.
Phytotoxicity, yeah, ma'am.
For
I
okay,
need to know.
I
Okay.
would like for botanicals, you can follow the Neem guidelines for the time being, but for each new insecticide, new guidelines are provided by the CIBRC. So, like for example, the Bolcure which I provided to you, it was a new molecule, new insecticide botanical which was developed by me. So at that time, there was no guidelines for the ball cure for the eucalyptus-based formulation. So what I did for the timing till the new guidelines was developed, I used the Neem as a standard guidelines, and at the same time, I requested the CIBRC with a proof that it has a insecticidal properties. It has got this one. I got it included in the schedule of the Insecticide Act. First thing, after getting included in the schedule of insecticide get, I requested the CIBRC to develop the guidelines. Okay, so that is the second step. And till that time, I followed the Neem guidelines. So whatever is the guidelines for Neem, I followed those guidelines. And till the time I got my specific guidelines for my molecule, I did the additional test which were prescribed on those ones. So like for five three tier toxicological tox is the simplest one. The earthworm, fish, and then human beings के लिए वो चार पांच हैं, dermal toxicity and skin irritation and so on. Inhalation studies, 3-4 studies are there that you can get it done just to find out कुछ problem तो नहीं है. So that you can get it done.
Thanks a lot, ma'am.
Thank you, ma'am. We have one more question.
Good evening, ma'am.
Good evening.
Ma'am, my question is that you develop a ball cure formulation. from eucalyptus that is specifically to cotton bollworm Helicoverpa armigera or other bollworm complex also, like
In
pink bollworm or
And no, it was not effective for the pig bollworm. It was only for the bollworm pig, that's Helicoverpa. But my American company to whom I gave, they tested against 50 more insects, and they found effective against 30 more insects. So we do not know the potential, full potential of this formulation. Okay,
ma'am, what is the LC50 value of that particular?
it was 0.05%. 0.05%.
0.05.
5%.
Thank you, ma'am.
Madam, just I want to ask one question more. Is there any ideal temperature and rainfall condition for highest oil content or acidic acid?
Now that I can highest, I don't have such idea, but only thing is it has the correlation with the rainfall. So as the
Yeah,
the
because
in
because in
if
Rajasthan,
the rainfall is low, the ASA will be
yeah,
low and the oil will be
yeah,
high. This is the only
yeah,
correlation which we got.
yeah,
Yeah.
yeah. As you have told that HRD is just inverse to oil content.
Yes.
So in Rajasthan, actually rainfall condition is also low and temperature is very high.
High, high,
So
high.
just we wanted to know about it that there should be some condition, ideal condition of temperature and rainfall, so that we can get high oil content and high HRD also. So there must be some condition, some optimum
We
situation.
can tell you the location from where we got the high oil content, and then
Yeah,
based on that we can find out like which are the conditions
yeah.
prevailing in those
We can
places, and based on which we can find out the places.
we can relate such.
Like I tell you now, the southern plateau is known for high answer. The
Hmm,
southern plateau, everybody knows. All the companies know.
yeah.
So if any collector is coming from the southern plateau bringing the seed, it gets very high price for the other content.
Definitely, their conditions are suitable for that.
Yes,
Yeah.
yes. So that area is now
Thank you.
known as a Neem area.
Yeah, yeah. Okay. Thank you, madam. Thank you.
नमस्कार मैडम जी।
नमस्कार सर।
मैडम हम लोग कुछ ही दिनों में 10-15 दिनों में सीड कलेक्शन के लिए फील्ड में जाएंगे। तो जब ये हमारे इधर निम्बोली बोलते हैं इसको, नीम के सीड,
हमारे यहाँ पे भी निम्बोली बोलते हैं।
तो निम्बोली निम्बोली मैडम है ना 4-5 दिन तक मतलब वो उसका पकने का वो चल रहा है ना, थोड़ी पहले पीले पड़ती है और उसको खाते हैं तो मीठा लगना शुरू हो, थोड़ा कड़वा होता है। फिर लास्ट में 5-6 दिन में वो पूरी पक जाती है और एकदम ऊपर से पीली हो जाती है। अच्छा, सुनाई दे रहा मैडम?
हाँ जी सर, मेरा फोन गिर गया।
हाँ, तो उसके बाद में उसकी जो पल्प है वो सिकुड़ जाती है, ठीक है, उसी पेड़ के ऊपर। और या तो सिकुड़ के वहीं रहती है या 4-5 दिन बाद में नीचे गिर जाती है। तो हमें ऊपर से सीड प्लेट करने हैं ताकि
ऊपर से येलो स्टेज पर।
हाँ,
ब्लैक
येलो
नहीं पड़ना
स्टेज।
चाहिए, ब्लैक नहीं पड़ना चाहिए।
नहीं, ब्लैक तो नहीं पड़ेगा मैडम, कुछ दिन है ना येलो भी क्लब सिकुड़ जाता है।
नहीं, सिकुड़ना नहीं चाहिए, सिकुड़ने से पहले ही आपको कलेक्ट करना है। सिकुड़ने से पहले कलेक्ट करना है। मैं बताती हूँ, डॉक्टर गुरदेव सिंह का जो है वो आपको येलो स्टेज में कलेक्ट करना है। इसको पानी में सोक करना है ताकि आपको म्यूसिलेज निकल जाए सारा। म्यूसिलेज जो है ना वो पानी, जैसे हम खरबूजे के बीज को सड़ाते हैं, इसको भी सड़ाते हैं पानी में, तो 2 दिन के लिए ताकि म्यूसिलेज निकल जाता है। इसके बाद वो म्यूसिलेज निकाल करके उसको छलनी में इसको
2 दिन में पल्प उतर जाता है।
पल्प उतर जाएगा। वो पल्प उतरने के बाद पानी में वॉश करना है। इसके बाद वो सैंड में रब कराते हैं, सैंड या राख जो भी मतलब। हाँ, सैंड से रब कराते हैं ताकि इसमें कोई पल्प ना, म्यूसिलेज चिपका ना रह जाए। वो म्यूसिलेज निकल जाएगा। इसके बाद आप ट्राई कर दीजिए।
सैंड में मैडम 2 दिन छाया में रखें।
हाँ, सैंड में 2 दिन। आप वो डॉक्टर गुरदेव सिंह से पूछिएगा उनका प्रोटोकॉल क्या कहता है। मुझे पता नहीं कि छाया में रखना है कि हाथ में, बट उनका कलेक्शन सबसे अच्छा था।
अच्छा, एक है ना मैडम, है ना ये जो सीड की साइज होती है वो डिफरेंट होती है, कहीं बड़े भी होते हैं, कुछ छोटे होते हैं। ठीक है, तो हमें कौन सी साइज लेनी चाहिए?
आपको तो सारा कलेक्ट करना जो भी आपके पेड़ पे आ रहा है, सेग्रीगेट नहीं करना उसको।
अच्छा, सब तरह का हो मतलब।
हाँ, आपको सेग्रीगेट नहीं करना।
थैंक यू वेरी मच मैडम, धन्यवाद मैडम।
एक्चुअली आप श्री कर्णाराम जी थे और हमारी टीम के इम्पोर्टेंट मेंबर हैं जो अभी सीड कलेक्शन चालू होगा उसमें।
हाँ जी।
और इनफैक्ट सीड कलेक्शन में हम लोग कोई सुपीरियरिटी को बेस नहीं बना रहे हैं, बिकॉज हमें सब तरह के सीड चाहिए तभी हम वेरिएबिलिटी स्टडी कर पाएंगे। इसलिए ये मैं इसीलिए अपनी टीम को बार बार ये बोलता हूँ कि ये एक ऐसा प्रोग्राम नहीं है जिसमें हमें सीपीटी सेलेक्ट करने हैं, हमें इंफीरियर भी सेलेक्ट करने, सुपीरियर पूरा रेंज आना चाहिए, तभी हमें पता लगेगा कि वेरिएबिलिटी क्या है एज अ डायरेक्ट इन कंटेंट की। तो उसी तरह से हम चल रहे हैं। इसके बाद मैं ऑनलाइन जो उपस्थित हमारे जितने भी श्रोता हैं उनको मैं इनवाइट करूंगा कि ऑनलाइन ऑडियंस में से अगर कोई क्वेश्चंस पूछना चाहते हैं, आई थिंक बोरा जी भी शायद जुड़े हुए हैं, बोरा जी अगर जानना चाहते हैं तो बोरा जी क्वेश्चंस पूछें आप।
नमस्ते मैडम, गुड इवनिंग।
नमस्कार सर।
It was a very informative lecture and I really enjoy because being a botanist I worked out on so many plants you have mentioned, right from the Anarica indica to the biodiesel of Jatropha plants, and besides Jatropha there are so many other biodiesel plants also. One more thing I just want to know that when you talk about the variability in Withenia sominifera, you have not mentioned the chemotype of Nagore. Is it any reason for that purpose?
नहीं, नागौर तो है ना, वो शॉर्ट टाइप जो नागौर का है ना, मंदसौर वाले कलेक्शन में है।
But in your chemotype mention, if you can see that you had Nagore was not mentioned because Nagore is known for within a sominifera.
वो एक्चुअली मंदसौर के कलेक्शन में था वो नागौर वाला।
मंदसौर is M45.
मंदसौर के जो रिसर्च स्टेशन था ना, वहाँ से हमने 160 जम प्लाज्म लिए थे, मतलब नहीं नॉट 160, अराउंड 50 जम प्लाज्म लिया था वहाँ से, टोटल 160 था, उसमें 50 मंदसौर से सारा जम प्लाज्म लिया था मंदसौर से।
जैसे कि आपने बताया
उसमें नागौर था,
शुरू में आपको बताया कि जो जोग्राफिकल कंडीशन, सॉइल कंडीशन, क्लाइमेटिक कंडीशन से काफी चेंज होता है।
हाँ जी।
तो ये विथेरिया सोमिनिफेरा कई जगह ट्राई किया गया।
हाँ।
तो ये जो कंटेंट, जो जहाँ पर बात करते हैं केमिकल कंटेंट की, तो काफी वेरिएशन होता है। और जो नागौर में जितना केमिकल मिलता है वो एल्सवेयर नहीं मिलता है। जैसे मंदसौर का
मेरा कहने का मतलब ये जो हम विजनोलाइट पे कंसंट्रेट कर रहे हैं, ये शायद गलत कर रहे हैं। मेरा कहने का अपना जो है, मेरा पर्सनल है ये व्यू, मेरा पर्सनल व्यू है जो मैंने स्टडी किया, क्योंकि रूट पे इतना विजनोलाइट होता नहीं है। विजनोलाइट की एक्टिविटी एंटी कैंसर वगैरह के लिए, इम्यूनोमोड्यूलेटरी के लिए नहीं है। हम लोग शायद गलत डायरेक्शन में जा रहे हैं विजनोलाइट्स के चक्कर में। आई थिंक जो पॉलीसैकेराइड्स हैं जो रूट में है वो इम्पोर्टेंट है इम्यूनोमोड्यूलेशन के लिए, और वो उसमें किसी ने स्टडी नहीं की है।
अच्छा,
ये मेरा समझ है। हाँ,
वगैरह जो है ये इतना इम्पोर्टेंट नहीं है,
इम्यूनोमोड्यूलेटर के लिए नहीं है। और अभी अगर आप रिसर्च पे देखेंगे तो आयुष ने भी मना कर दिया है विजनोलाइट के लिए। आयुष ने भी इसको क्लियर कट गाइडलाइंस दिए कि विजनोलाइट को कंसीडर ना किया जाए।
जैसे बारबेरिस की बात की है,
ये सब चीजें आपको तभी पता लगेंगी ना जब आप इन्वेस्टिगेशन करेंगे। जब इन्वेस्टिगेशन होगा ही नहीं, केमिस्ट्री करेंगे नहीं, कुछ करेंगे नहीं, तो पता ही नहीं लगेगा। आप वही करते चले आएंगे जो आपको बोला चले जा रहा है। लेकिन जब करके देखते हैं तो आप कुछ और पाते हैं जब आप करते हैं प्रैक्टिकली।
जैसे हर जगह बात होती है कि जैसे रूट में अगर कोई कंटेंट है केमिकल, तो वी आर गोइंग फॉर नॉन रेस्ट्रिक्टिव हार्वेस्टेशन बिकॉज when we take out the root then it will again difficult to raise plantation again.
ये वो लीफ को प्रमोट कर रहे थे ना, लेकिन लीफ में तो केवल विजनोलाइट है, पॉलीसैकेराइड तो नहीं है ना। हमारे आयुर्वेद में जो है रूट को प्रमोट किया गया है, हमारे ड्रग्स जो है रूट को। तो वेस्टर्न कंट्री ने केवल लीफ को फोकस करके वहां से विजनोलाइट ला दिए, लेकिन अगर हम देखें तो विजनोलाइट कंटेंट तो बहुत ही कम है रूट में।
जैसे विथेनिया का एक और प्लांट है, विथेनिया कोगुलेंस, पनीर बंद जिसको बोलते हैं, वो हमारे उधर काफी है।
वो पाकिस्तान का प्लांट है वो जो कोगुलेंस।
वो मैडम इंडिया में राजस्थान में कुछ था मैडम, बाड़मेर रीजन में खूब था
प्लांट है ना उधर।
पूरा राजस्थान पाकिस्तान बॉर्डर के था काफी प्लांट।
पाकिस्तान में डोमिनेटिंग है कोगुलेंस जो है, वहां से सोमनिफेरा नहीं है इतना।
उसमें भी काफी सारे अच्छे केमिकल कंटेंट थे जो कि स्पेशली फ्रूट में, जिससे कि आपका पनीर बनता था। दूसरा एक बारबेरिस की बात की आपने मैडम, बारबेरिस यू टॉक अबाउट द बारबेरिस वल्गेरिस और एक्विफोलियस।
मैंने बारबेरिस की सारी स्पीशीज बताई थी ना, एरिस्टेटा भी है, सारे 7 स्पीशीज कलेक्ट की थी, 7 स्पीशीज कलेक्ट की थी।
काफी सारी स्पीशीज इसके हार्मफुल इफेक्ट काफी ज्यादा है, साइड इफेक्ट बहुत सारे हैं।
नहीं, एरिस्टेटा है ना, रिकमेंडेड तो एरिस्टेटा है।
हाँ।
आयुर्वेदा में जो रिकमेंडेड है वो एरिस्टेटा है, लेकिन सब्स्टिट्यूट यूज करते हैं लोग बाग उसका।
सीबकथॉर्न की बात की मैडम, जो आपका हिपोफी रेगुलरिटीज जो है आपका उत्तराखंड का,
हाँ जी,
तो इसके तो काफी सारे अभी और भी नए केमिकल निकल रहे हैं, कई सारे नए यूज हो रहे हैं इसके।
हाँ जी,
तो ये प्लांट को काफी इम्पोर्टेंस गेन कर रहा है, तो इस पे और रिसर्च होना चाहिए।
बिल्कुल
आपके
होना चाहिए,
एमपी
बिल्कुल
में
होना
हो
चाहिए।
रहा है।
आप आइए साथ में, हम बिल्कुल करेंगे।
थैंक यू मैडम, थैंक यू।
अच्छा,
वेलकम।
ये जो आपने म्यूसिलेज की बात की ना डॉक्टर तरुण, इस म्यूसिलेज से what sugar content is very high, and it has been used for development, for converting into a wine or beer बनाई है उससे, म्यूसिलेज से। जो नीम का
आप
जो
जो एमिटी फाउंडेशन आपका काम कर रही है, एग्रीकल्चर के ऊपर भी काफी काम कर रही है।
हाँ जी,
और हम लोग जो है आफरी में सैंडल का एग्रो फॉरेस्ट्री बेस्ड प्लांटेशन कर रहे हैं।
वेरी गुड सर।
So it is a new approach actually, so that farmers can also get the income as
हमारे
a
पास
sandal.
एग्रीवोल्टेक भी है, वो सोलर और उसका दोनों कॉम्बिनेशन ना।
और सैंडल आजकल आपको पता है कि स्किन और कैंसर वगैरह में भी काफी काम आने लगे हैं सैंडल के प्रोडक्ट जो है।
हाँ जी, हाँ।
तो इसमें भी अच्छा काम हो सकता है। सो आई थिंक तरुण कांत सर इज अवर जीसीआर सर, सो वी कैन हैव सम collaborative work of this type of things.
मैंने तरुण जी को लिखा है उसके लिए अभी एफसीसी के लिए ना।
जी, yeah. Definitely, ma'am. Actually, I hope in future we'll have more association with you, with your expertise, and many more projects. There are a lot of things going on, and we see that we can have, I mean, your guidance in many senses. So we'll explore those possibility. Yeah, thank you, Boraji. Thanks a lot.
Thank
And
you.
is there anybody else from the audience, from the online audience, who would like to ask? We can have one quick question.
Good evening, sir. I have one question, sir.
Yeah, good evening, Desha. Good evening.
Good evening, ma'am.
Good evening.
Ma'am, I just want to ask one thing, that after harvesting seeds, कितने समय के लिए वो हमारे मतलब आपकी लैब तक आ जाना चाहिए, so that कि उसमें
as soon as possible, as soon as possible.
Okay. कोई मतलब लिमिटेशंस के इतने एटलीस्ट इतने दिन में तो आ जाना चाहिए, इफ मतलब कभी ऐसा हो कि हम लोग नहीं शिप कर पाए।
मैक्सिमम विद इन अ वीक, मैक्सिमम विद इन अ वीक,
विद इन अ वीक में शिप हो जाना चाहिए।
Actually, ma'am,
I would suggest कि पर्सनली कोई लेके आए तो that's much better rather than शिप में कई बारी प्रॉब्लम्स हो जाती है शिपिंग में।
ओके, थैंक यू मैम।
Yeah. In fact, ma'am, I'll just supplementing what Desha said. I would also like to ask you one thing, that because our area is so huge, and particularly in some other parts, like for example, in Maharashtra, the team in Maharashtra, they say that they have got a very widespread collection, and for them to get the seeds and then process them and then send them, it is taking a little longer. So what we have also done is we are also trying to document this time. In this our seed collection, we have prepared a format which is being given to all our partner institutes, that the time of collection, time of processing will be documenting each and every day, so that we also have the data on how much delay is there, so that we can correlate later on with
meanwhile they can store the seed in fridge के अंदर से store करके रख सकते हैं, in box,
okay.
in plastic boxes they can keep inside the fridge.
Okay, because ma'am, it will be coming in like it cannot be done in one day. There are 100 samples
Till the time they are
distributed
processing
around.
rest of the seed, जैसे आपका आज प्रोसेस हो गया, ड्राई हो गया, you are absolutely sure ड्राई होगा, उसको बैग्स पे पैक करके you just put it inside the fridge.
I mean, do you think, ma'am, fridge means 4 degrees or minus 20 or
4 to 10, between 4 to 10, anything.
Okay, just normal fridge.
Normal fridge, not deep freezer, not deep freezer. 10 degrees
not
is
the freezer, not
up to 10 degrees is good enough.
Okay. And what we were planning was that we were planning to keep sending you the seeds as and when they get collected. As soon as we have say some 10 or 15 samples ready with us, we'll ship it to you. That's what our plan was.
ठीक है, मैं अपने आप पर फ्रिज में रख लूंगी. Is as good as this thing, so that's not an issue.
Okay. All right.
Not an issue.
मैम,
yes, Desha, you were saying something.
Yes sir, I was saying कि अगर हम उसको स्टोर करते हैं फ्रिज में तो कोई वेरिएशन नहीं आएगा, लाइक कुछ जो पहले से स्टोर्ड है कुछ फ्रेश आ रहे हैं?
नहीं ऐसा कुछ। आपको डेट ऑफ कलेक्शन तो लिखेंगे ना, date of harvest
हाँ जी
technique.
मैम ओके थैंक यू मैम
Okay. Okay. Right. Yeah. So, right. Anybody else from the online audience? Okay. It looks like there is nobody else. So, yeah, mute. Mute. Yeah. Can you bring ma'am का screen लेके आना? Just pin ma'am. निखिल मैम को ये कोई स्क्रीन आ रहा है एक नहीं इसको नहीं बंद करो इसको ये पे अनपिन करना है अनपिन करो या चेंज द लेआउट गो टू लेआउट एंड चेंज टू स्पॉटलाइट है ना ऑटो कर दो जस्ट डू इट ऑटो या नाउ फाइन या सो मैम it was really very enriching in fact deliberations that have taken place today. In fact, your talk was so thought provoking that a lot of people have asked so many questions. Many times it happens that in a seminar at the end there are no questions. I mean, I sometimes feel that the number of questions asked in fact define the kind of presentation that went on. So I mean, congratulations to you and to all the audience for having witnessed a beautiful presentation which was very scientifically very enriching, and we have come to know a lot of things about secondary metabolites. We are also working, but there were many new things, a lot of learnings that have taken place, and we still have a lot of questions. And I, in fact, insist others who may have questions may also write emails to ma'am, and she can always reply you back. And it's now time to, in fact, end this today's seminar. And at the end, as it is customary, it's my duty to propose a formal vote of thanks. And at the very outset, let me first of all thank our director, sir, to in fact have said yes, and in fact have when it came, your name came up for this, and he really wanted to be here today, and he was talking to me today morning in the phone, and he said that he will missing out on this, and so thanks to him for giving us this chance to have you with us, and thanks to you for in fact agreeing to give this lecture, and for in fact coming up with such a nice lecture which we all have we really feel so enriched today. And thanks to all the participants who have joined online as well as the offline participants sitting here. And I also thank the IT cell people for arranging this hybrid mode webinar today, as well as the GCR office people for arranging the logistics of this today's meet. Thanks to all. Thank you, ma'am, once again. Thank you. Thank you.
Thank you, Tarunji, for this opportunity, giving me this opportunity for presentation. Thank you so much.
Thank you.
Thank you.